really off the slope of this little cone, so it should be fairly flat here, I would think.
Yeah.
Yeah, it looks that way from our nav plot.
But one of the things I saw in Nav when he had pulled out before were actually there are 3 of these cones, and we're kind of diving between 2 cones, and so I imagine it's kind of a constricted space, so perhaps water may be funneled through here and you get a stronger current than you might expect otherwise on the surface.
Yeah, that could be. We've seen that, I think, at lots of scales here as we've been on this expedition, so it's a little different look to the bottom here.
Yeah, very.
Yeah. Huh.
So this is different than most things we've seen so far in the dive, and that's always fun. It's nice to see the different kinds of geology. I already see a fish down there. Looks like maybe a, well, I'm not going to guess. Oh, what the heck? It looks like a little
I'm going to
halosaur.
speculate that it's a halosaur, a deep sea eel relative, the group called spiny eels, but we need to get a closer look, of course. We often see these when we first settle down at these depths.
Great, thanks, Bruce. We're still settling in, so we're not quite ready to do our close-ups, but we'll let you know.
Yeah, I think it was Astrid that made an interesting observation yesterday. Is we often see fish when we first drop down in the water, and then we don't see that many as we start to move, and we wonder if there's avoidance of the vehicle by the fishes. That when the vehicle first drops down, it's happening too quickly for them to scoot away. But this is a good example of how we very often see a fish or two as we first approach the bottom. You can see the long whip-like tail, and you can also see a short little fin sticking up about a quarter of the way back on the fish. It has a flattened snout, so this is definitely a halosaur.
Bruce, I don't feel like...
in on the head to get a idea. I think it's an Aldrovandia. It looks like the head is fairly translucent and unscaled, so this would make it Aldrovandia, which is the genus that we see most often.
Bruce, I don't feel like we've seen all that many halosaurs on this expedition. Am I just forgetting, or is that accurate?
No, actually, I think you're right. I think that those that we have seen have been early in the dives, on some of the deeper dives, and we haven't seen as many on this expedition as we've seen in the Marianas and in the Hawaiian archipelago in the past couple of years.
Thanks, Bruce. I noticed as we're following the fish that there are a couple of corals that were growing off of this substrate. And it's kind of interesting because it's so flat, and yet they're coming right out of there. That's got to say something about the currents coming through here.
You're right, yeah. Now that we're zoomed in a little more, I think what we're seeing is this manganese crust, this fine texture. And then just, like you said, we first sat down, just really a fine dusting of sediment across this. So there should be plenty of attachment places for sessile organisms.
I see the lasers just passed over an urchin, and I'm seeing lots of these small yellow sea fans down there. And Chris Kelly earlier had said we might be a little bit too deep for precious corals, but he's expecting to see plexaurid corals. Is there a larger fish? There's a larger fish at the top of the screen, or is that just a, no, sorry, my bad, pilot, it's a smudge. Yeah, this is a different fish. I was looking at something different. Oh, I wasn't crazy. It is a large fish there. I thought I saw something.
Start with the big guy, yeah.
Oh, wow. Okay, I'll have to log these in a bit. That's an oil fish. It's a type of snake mackerel. It's actually a commercially fished species. The species name is Ruvetus prediosus. The mouth's full of very large canine teeth, so this is one of the top predators in this ecosystem. The flesh is very oily, hence the common name. They're edible. But they also have a sort of purgative, laxative effect if you eat too much of the fish because of the high oil content. So that's an amazing thing to see down here. And the other fish, there's a fish at the top, too, that's another halosaur.
Yep.
And then the other dark fish that we saw was a cusk eel in the genus Lamprogrammus. So I have lots to log in our data logging program and in the chat room.
Great. And pilot, I've seen a number of yellow fans when you get into a good position. Just choose one and let's have a look at it. There's several of them. The ones on the right on that exposed rock, there might be 2 different kinds. So I know I told you any one, but no, sorry about that. That is a hermit crab, I believe, with a zoanthid on its shell. Yes, indeed.
All right.
So pegurid hermit crab, you can see its legs sticking out and its large yellow claws on the left side, its antennae. And then that sort of oily brown thing that you see on its back, that is a zoanthid colony, colonial anemone. And you can see 5 of the polyps extended, or 6. And I believe this, or maybe 7, it might be classified in the genus Epizoanthus. So this is a species of zoanthid that does live in association with these crabs. It's not an unusual observation to make here, down here in the deep sea. And the presumption is that the zoanthid is protecting the crab to some degree from predators, and the zoanthid obtains food as the crab is moving around. Thank you, pilot.
Looks a little heavy to carry around, but I see its purpose.
Yep.
Hi, Scott.
Yeah, that's a good one.
So this looks very similar to something that we have in Hawaii. It was identified as Sympagurus burkenrodsii. The crab that's what the crab is. And then, yeah, I think your idea on the zoanthid was as far as anyone could go, but it basically is a dead ringer. There's some pictures in the Hurl Animal Guide for this because it was imaged back in 2003, I think. And it's the first time we've seen one of these things outside of Hawaii. So this is very, very cool. But yeah, I think it's Sympagurus is the actual genus for the crabs that carry these things around.
Thank you, Bruce.
Oh, that was Chris.
Thank you, pilot.
I'm not sure if the fish on the right is a different species. I think that's a fish. Maybe it's just a dark streak on the rock. But
Yeah,
the
Bruce.
one up in the center looks like it could be another lamprogram. Oh, no, that's a shark, a little shark. That's a great find. I'm not very good at identifying sharks, unfortunately, so I can't say what this might be. It could be one of what we call the green-eye sharks in Hawaii. But maybe Chris has a better idea.
Might be a rat tail behind it as well. Go ahead, Chris.
I'm looking at it, Bruce. Not really sure. I mean, I think you would have the best idea on this. The squared-off caudal fin would rule out perhaps the squalidae. Is that correct, Bruce?
You know, it's not a squalid. They're more slender-bodied and lighter in color. Unfortunately, there are a number of these that, there's a macrurid behind it, too, that could be a Coryphonoides. This is a very fish-rich site. So I don't know if it would be something like Centrosymnus or Centrocilium or, I don't know, it doesn't look like a Latius. It's a squaliform shark in the dogfish order. They've been split up into several different families. It's not a lantern shark. It's not a Netmopterus. Have to send these images out for better identification, I think.
Thank you, pilot. Yeah, there's plenty of stuff on the bottom here. So we're getting a zoom in here on a sea fan in the family. Well, I'm going to hold off on the family. I was going to say Plexoridae. It could actually be an Acanthogorgiid. Little bit hard to tell. This one you can see has lost a lot of tissue, and I've seen several of these colonies in the area. You notice this is on a little cobble. This is maybe interesting because it looks like it's tipped over, which would suggest that it's loose.
Yeah.
You know? It's growing sideways instead of just directly up, and I wonder if that's part of why it's in such bad shape. And so one of the things that happens is the larva settles on the rock, glues itself to the rock, and then begins to grow into this colony form. And it's a fan sticking up in the current. And if it doesn't have a good, stable rock that it's on, something like this happen, it tips over. But then you see it can kind of reorient its growth to grow upward. So that's a possibility of what happened there. The other possibility is that cemented in place and it just grew sideways.
Yeah, these rocks really look like they're crusted in there.
They sure do.
So, but who knows?
But one thing I wanted to note, I've seen several of them now. They're fairly small, and so far the ones that I've seen, none of them have a brittle star in their arms, and you would often find brittle stars in the arms of these sorts of colonies.
So Ken Sulak, who's in Florida and knows a lot about deep-sea fish, says that shark was in the genus Centrocilium, and he put that into the chat room.
Thank you, Bruce, and thank you, Ken. So you can continue to see some of these really small yellow sea fan colonies dotting the bottom. I noticed before an aspidodiademid urchin on the bottom.
And again here, the bottom substrate is this, we think, manganese crusted rock. And then we're seeing just really a thin dusting of sediment across it. So this is a little bit different than some of what we've seen before. There's really this fine texture to the crust, and then we've got the sediment sort of dispersed around it.
I see another halosaurus swimming through. Image one of those before, and I can't tell if there's something kind of white in the back distance. No, there's something right on the bottom. It's coming into the center of the screen now. It's in the distance. It might just be a little bit edge of a rock or something. Oh yeah, it looks like it's a big rock, and then there's some winnowing of sediment at the base, I see. You can see now lots more of these colonies, lots of these yellow sea fans.
Yeah, I can certainly see everything moving off to the right side of the screen here with the current and that sediment there on the lee side of that boulder.
It's funny how this can be.
What did we decide the sediments we're doing here, or what's the substrate?
Yeah, if we were zoomed in, we could see clearly. It looks like these are a manganese crust on the rocks, and it looks a little different than some of the other places because the sediment is actually filling in that fine texture that develops on the manganese crust, I think. And so when we're zoomed out, it looks a little different than many of the other sites we've visited.
Yeah, it's very, very deceptive. You might think it was a jumble of cobble or rocks, but in fact, it looks like a pretty solid pavement. So we're looking at a couple of things here. An echinoderm on the right. This is, I've been told, has been Plesiodiadema would possibly be the genus name for this urchin with the long curved spines. And then you can see this thread-legged shrimp to the upper left, Nematocarcinidae. And then if we look at the coral, you can choose one direction or another, left or right. I think they were on both sides. Again, we've been noting these are very abundant. This one doesn't seem to be damaged at all. You can see pretty full polyps. There's a little anemone at its base. And again, I'm not really comfortable at this point whether I can say that that's in the family Plexauridae or the family Acanthogorgiidae. Both I would expect to find here. Both have this sort of general sea fan appearances, but there are some detailed differences in the shape of the polyps and then in the type of sclerites that they have. Sclerites are these calcitic elements, microscopic in size, that are embedded in their tissue. And I noticed perhaps a small cup coral down there as well. Thank you, pilot. James Aubry is in the chat room. He's a graduate student at the University of Louisiana at Lafayette. He's a PhD student with me, and he notes that these small colonies are not unlike acanthogorgid specimens in the Paris Museum that were dredged from the Mozambique Channel. So then it's likely that those are acanthogorgiidae. Thank you, James. We're now looking at a rock pen. This is another kind of octocoral, and we've seen not a huge number of sea pens, but we have seen some, and sea pens are adapted to live in sediments, but this is a great view of one that has changed things up a bit. That swollen structure you're seeing at the base is called a peduncle. That's usually inflated and buried in sediment and gives it anchorage in sediments. But here, this group of sea pens, and there's several species that have shown this morphology, is using it more like a plunger or a suction cup to attach to a rock. And that suggests to me that there are not enough sedimented areas for it to be deep enough to anchor into. And this may be in the genus Calibelemnon. They're kind of hard to determine. And in fact, in that genus, one of the things about it is it's got relatively few sclerites. It's kind of interesting. We're just talking about sclerites being really important for identification of octocorals, so when they don't have them...
The absence is also important.
Important, and then it makes it hard to compare and identify. So our depth is 1,029 meters, and we've just begun our dive on this cone-like feature. We're actually at the base. We're not actually on the cone yet. The cone is to our northwest. We're heading in that direction. That's the direction that the camera is pointed right now. We're going to go upslope. And we're just offshore of Kingman Reef. And we're in the Pacific Remote Islands Marine National Monument, the Kingman Reef and Palmyra Atoll unit. So this is the first ever view of the communities and the geology at this depth in this monument. Or perhaps I should say this unit of the monument, since we've had other dives in other parts of the monument, different units. So, Del, what was the bottom here when it's becoming manganese coated? I'm trying to picture. Was it smooth? Was it a bunch of rubble that gets kind of glued together?
Yeah, that's a good question. So this little cone we're on seems to be fairly peaked, but it is at a shallow depth. So yeah, one of the questions would be, did it have some sort of carbonate material developed over it, or was it volcanic material that would have formed when the initial volcanic platform here formed? So it's a little hard to tell. This pattern that we're seeing, these little kind of look like cobbles, I suspect are just the pattern that we've seen over and over again on these manganese crusts. So it's a little difficult to say for sure if this was rubble or what.
So we're getting a close-up view of an urchin. And Chris, if you want to say something more about this urchin, you were talking about it yesterday at Diademid, and I think you said that it might be a coral predator, and I see this one little coral next to it, probably a smaller stage of the colonies that we've been looking at. And I wonder even if these sorts of urchins can eat the young recruits.
Yeah, well, if I was that little coral, I might be terrified right now, but who knows what, the feelings that corals have. But anyway, this is a pencil urchin in the family Sideridae, and the one yesterday, because of the depth and its general appearance, I thought it could be Histosideris, and I think this could possibly be the same thing. I've never seen that urchin so close up, and maybe Chris Ma, if he's listening, he'll chime in on this, but that's what I think it is, maybe in the genus Histosideris. And from what we surmise right now and all the observations we made of these urchins, they are found on corals a lot, and they're very likely feeding on them. So if I was that little yellow coral, I'd be very afraid.
Interesting, and I imagine that's a relatively old urchin, given all the stuff I saw growing on some of its spines. I saw a number of hydroids, for example, that had settled on the spines and begun to grow.
Yeah, I'll just note that urchin had mostly white spines, but there were two shorter red spines sticking straight up from it like devil's horns. So, interesting.
Yeah, I noticed there was some tissue on the tip of one of the spines, which suggests that, it's just been scraped off of other parts. Here's a different kind of urchin. This might be in a kind of third urchin. what I usually call the pancake urchin. Maybe it's tam o'shanter, or would have thicker bases to the spines. Certainly see the color is different. You can see some of those spines, the tips are kind of bulbous. There's tissue on them, and I know from experience that some of those can pack a bit of a toxin. We have collected one of these, or I have been on a cruise where we collected one of these from a seamount off of Hawaii, and pulling it out of the bio box, I had the misfortune of getting one of those spines into my knuckle, and that burned for a couple of days. So pretty potent, and probably a very good defense against predators.
That's why you never suggest we collect them.
I've learned from my past mistakes. And you can kind of see at the base of those spines, there are these knobs, and there are articulation points, and you can see the spines are moving around. There's little bits of muscle at the bottom there at the articulation, and so they can control the direction that the spines move, and then you see the waving tube feet. Thank you, pilot. Those two laser dots that you're seeing are 10 centimeters apart. We use them for scale, so that tells us that urchin is maybe 11 centimeters in diameter. So one thing that's interesting here as I look at all these yellow fan corals, they're all really small, which suggests they're relatively young. I don't know how fast they grow in this particular area, but it makes me wonder why there aren't larger ones. And that might say something about the conditions here, how recent it was that the bottom has been exposed. Maybe the currents have changed and washed away some sediments, or there's been some other events here that have prevented growth over a very long term, so you don't get really large fans. I noticed the one that's up on the high point is bigger.
A little bigger. Yeah. So we're gonna get a look here.
Yeah, what do you think, Del? What's this going to be?
I don't know. I think it's going to be a sea star here.
I think you're right. I wonder if it's draped over something that it's feeding on, or it's just kind of hanging out on top of this rock. Kind of looks like there's a lot of current scouring here. Might be the base of a larger coral on the right side, or maybe that was a barnacle, large barnacle. You can see all the tube feet kind of hanging out on the underside of these arms.
This guy's all legs here, it looks like.
This guy is all legs. I'm not sure if this is a Henrycia type. Chris Kelly, do you want to let us know what you think?
Yeah, because when I butcher this, then that'll prompt Chris Maw to call in.
There you go.
That's what we like to really hear. But I was originally thinking this was Henrycia, but now with the close-up, I don't think so. I think it could be a Zoroasterid in the genus Zoroaster, excuse me. But hopefully Chris has taken a look at this. I've never seen one draped on a rock like this. Most of the time you see them out on the sediment, but those extremely long arms, I guess somebody thought they resembled swords or something like that. Zorro. That's probably completely wrong, but that's the way I remember these guys. And anyway, Zoroaster is my best guess on this one. Now call in, Chris, and correct it.
I think we're seeing a stoloniferous octocoral here. I just wanted to get a brief look, and sure enough, you see that little ribbon of yellow. There's 1, 2, 3, 4, 5, 6, 7 polyps in a row. That's something that's called a stoloniferous octocoral. Grows in this ribbon-like manner, can make a mat. It doesn't produce an upright skeleton. Thank you. And then you can see 1, 2, 3, 4, 5 or so small sea anemones. A couple of them completely contracted, so you can't see their tentacles, and the others with their tentacles out. Thank you. Nice observation.
Chris Ma here.
Hey, Chris. It worked.
Yes, Chris Kelly is always good at getting my attention. Unfortunately, fortunately in this case, it's also unnecessary because he was right. The sea star you were looking at is most likely Zoroaster. They're a fairly commonly encountered group of deep sea sea stars. These are actually considered kind of basal in the larger group of what's called precipulate sea stars. There are a fossil record of these animals that occurs back to the Jurassic, not Zoroaster specifically, but Zoroasterids. And these are usually, it's interesting to see them draped over a rock like that, because usually you see them, at least in my experience so far, usually you see them on sediment, sort of digging into the mud and sand, trying to get little mollusks and other things to eat. So I'm not entirely sure what it's doing up there, but maybe there's something that's trying to get to or whatever. I think I read once that some of the carbon isotope studies or whatever it was said that they might get some of their nutrients from phytoplankton or something like that. So maybe it gets food from another source by raising up, getting into the water column. I'm not sure. But I will say this, they look relatively innocuous, but the very fine spines on the surface are needle sharp. And even years after they've been sitting in jars and preservatives, I've been nailed by those things. They're not something you forget. They're not they're not poisonous, but they're very, very pointy.
That's two straight
Anyway,
echinoderm stabbing scientist stories. That's great.
Don't study echinoderms.
Yes,
What have we learned?
exactly.
Yeah.
Thanks,
Oh, yeah.
Chris. That was excellent.
And it was very interesting for Bruce also to mention on that pteroid those two kind of devil's horn spines. I was thinking that Chris was right, that it was probably something similar to Histopsidoris. And then I noticed those two, and I don't know I've ever seen those before. That's very interesting. So I'm going to go have to find out more about that at some point. But seeing material grown on the spines, I don't know that it didn't look like there was a lot on there. So because they don't have skin on the spines, so anything can grow. And I don't know if some of them may cultivate that, or if some of them just let it go, or it's not like a crab thing where they shed their skeleton. So there's definitely sort of a different dynamic than what you get with things like crustaceans and so forth. But anyway,
Well, Chris,
good start to the dive. Looking forward to more.
Let me ask you, Chris, do the spines normally have tissue covering them initially? Because that's what I thought I was seeing, tissue at the tip where it was reddish, but stripped down towards the base, and then many of the other spines just were without tissue. And let me just say for the audience that we're looking right now at a hydrozoan colony, which is really nice because we don't often get a really good view like this. Go ahead, Chris.
Oh, sure. So most of the time when you see sea urchins or any echinoderm, even the spines are covered with epidermis. So, if you look at a spidodiadematid or a diadematid or any, usually you're going to see skin all the way to the tip, and, it's there, you just can't see it. There's a ciliated epidermis, as your invertebrate zoology, tests, I'm sure will remind you. But pteroids are really weird because they don't have skin on the spines.
Oh.
And so, I think I've never actually tested it, but I think it ends or it sort of halts right at the base there where the color changes. So pteroids, for the general, as far as I know, I don't know if there are any pteroids that have skin on the spines, but that's sort of that dynamic is when you see the spines without, when you see the spines covered with epizoites, barnacles and hydroids and things, that's because they're growing directly onto the skeleton. So they're kind of weird in that way. And then, of course, they're shaped strangely in some of the Atlantic and other species in the South Pacific. They're kind of pencil-like in Pisciceterus and some of these other things, but there's a couple of them that literally look like umbrellas. They kind of branch out and form kind of upside down, curved, crazy structures. Some of them are almost surreal. It's like if Dolly had created a sea urchin. It's very bizarre.
Got to interrupt you there, Chris. We're looking at a fish, so thanks for the comments, and we'll hear from you later.
Happy to hear it. Good going today. Bye-bye.
Thanks. Bruce, I think this is an Oreo.
Yes, you're right. This is an Oreo dory. They get that name from the family name Oreosomatidae. Oreo is mountain, soma is body, because the juvenile stages have large conical scales on the body that the adults don't have. So they're not named after the cookie. But I've said before on these dives that this was a family that was unknown from the Central Pacific before 2005, and we're seeing them at these shallow dives almost everywhere. It's the genus Neocytus. It has little spines on the snout and above the eye that are supposedly a diagnostic character for a species in the western Indian Ocean, Neocytus acanthorhynchus. Acantho is spine, rhynchus is snout. They're related to the John dories that occur in more shallow water in the North Atlantic and other places. There are other larger species of Oreos that are part of the commercial catch in deep water fisheries down around New Zealand and Australia. They were caught by the orange roughy fisheries that were not sustainable, so that have been shut down. It's an Oreo dory.
Thank you, Bruce.
Yeah, Scott, this is Chris Kelly. I just wanted to make a comment for you to be on the lookout for this. And I'm not sure it was noticed by the pilots. We were so focused on the sea star, that Zoroaster, but right next to him on the rock was one of those giant barnacles, the Striatobalanus in the family Archaeobalanidae, I believe, and I saw another one as well. They look like dead coral bases. But maybe these big white things you see coming up here could be some more of these really giant barnacles. They're quite interesting, I guess, if you like barnacles, which I do. But anyway, if you have a chance to zoom in on one of them just to confirm that, that'd be great, even though there's a huge coral behind it. But
yeah, I was going to say, speaking of giant, yeah, Chris, I did see that. It was a barnacle. I'm not sure that it was still alive, but it was a really good size. We will definitely keep our eye out for them. As Chris noted, we have a very big coral fan coming up here in the upper right. I see another rock pen down below, some more of the yellow, what we're believing is acanthogorgiid fans. And actually, the acanthogorgiid is interesting. The rock pen's okay. We got a good view of that before. And we're okay with the yellow fan. We got that before. I see a cup coral in the background. But as we go towards that giant coral, and I realize you're at the end of the tether, you're waiting, further to the left, there was a white coral before that may be something different. Thank you. I was going to say that, so James Aubrey had identified these as possible acanthogorgiids, and that would be interesting because the acanthogorgiids don't have those large Ophiuroids, the brittle stars in our arms, like all the plexaurids do, or many of the plexaurids do. So that would make total sense. That would support that.
Yeah, we certainly haven't seen any of those yet. So,
exactly. So I think we're seeing mainly down on this flat area a forest of little acanthogorgia. So it'll be interesting as we transition to the slope. Do we still see those? Do we see something different? And already here you can see there's two different colonies. So I think this one in the foreground that's kind of a pink-white is going to be a primnoid in the family Primnoidae. So another kind of octocoral sea fan. It's a very diverse group. There's some 3,000 species of octocorals, so we're only touching on a few of them. They're really diverse in the deep sea. And I'm very excited. That's a bamboo coral in the background. Can't wait to get to that one. That's my specialty. But let's look at this one by the lasers first. All good things come to those who wait,
That's
though.
right. Oh, there we go.
Yeah, Chris Kelly, what do you think? Narella, it sort of has a classic Narella shape to it. And by that I mean it's a slight bit of a bush. You can see it's quite rigid, and notice those polyps are kind of oriented slightly downwards, facing downwards.
Yeah, Scott, I would agree. I would even say that it looks like Narella dichotoma with the bare base right there, and just the way the branches are coming up. We can't really see whether it's dichotomous branching, but it looks like that. The only other thing I think it could be that we know of right off the bat that look like this could actually be Calyptrophora wyvili, which also has downward facing polyps, but I don't think so. I think better guess is Narella dichotoma, so I agree with you.
Thank you, Chris. And so Chris mentioned two possible names there, Narella dichotoma as the species name, or the genus Calyptrophora, and both of those have polyps that are facing downwards, and one of the things that we would need to see is some detail on the sclerids themselves, the scales. And you know, Chris, as I look at this doesn't actually have a feel of Narella's scales, so maybe it is a Calyptrophora, but we can study the detailed images a little bit later. Another thing I thought was interesting, Chris noted the base, so where it attaches to the rock, that's called the holdfast. And if you look at it closely as we go out here, you'll see there's living tissue that's kind of pinkish in color that stops at a certain point, and then you'll see more of the white exposed holdfast. There you go. It's kind of whitish yellow at the base there. So that's the protein skeleton with a little bit of calcium carbonate embedded with it, and then the live tissue is that slightly pinkish area. Thank you, Pilot.
Yeah, we're seeing some of these bigger boulders here. It looks like at the bottom of the slope now. Almost certainly this doesn't look to be in place. Looks like things that have come down. And that's creating a little more topography. You can see some thicker areas of sediment here. So potentially we'll see different animals as we move up here.
Yeah, I was going to say, that's interesting. We didn't see the sediments in that open part, but they're kind of tucked in here, which I think maybe further supports that you might have a good strong current sort of being pushed between these two cones that are next to one another.
Right.
So.
Yeah, you can see the top parts of these boulders are still swept clean. It's really the low areas here and the lee sides that are getting the sediment.
So again, you see in the center of the screen on the sediments now two red dots. Those are lasers. Those are 10 centimeters apart. So as we come around to this coral, you can all try to help me measure the size of it. How big is this thing? And I'm going to tell you it's pretty big. And it's got a number of different things on it.
Yeah.
Lots of coral colonies here. There's another of the Oreo fish and a different bamboo coral whip behind it. As the fish swim, I'm always looking behind the fish while my fish colleagues are looking at the fish. This is great. It's interesting, you know, so we've talked a lot about how other animals in the community will use the sponges, will use the corals as a habitat of their own. That is, they're climbing up on them. And we've pointed out that most of these yellow fans don't have anything in their arms. So that's kind of interesting. You wonder, is there some secondary metabolite, some chemical that those corals are producing that wards off things from settling on it, or is it something about how spiny acanthogorgiids are? Well, as I look at this bamboo coral, there's a couple of things on it, but there's not much.
Not a lot. Yeah, I was going to expect more when we got close in.
Yeah. And that's not unusual for bamboo corals. They produce a lot of mucus, and I just wonder if there's some chemical that's in that mucus that when the larvae of these things settle, or even when they're adults, there's something about it that is distasteful, or they don't appreciate in some way. All right, so let's see those lasers, 10 centimeters across. So 1, 2, 3, 4, 5, 6, 7, 8, 9. That's at least a meter across in its widest dimension. So just over 3 feet wide, and it's a little bit taller than that. And I'm going to guess that this might be a colony of Isidella, something close to Isidella trichotoma. A bamboo coral is so named because its skeleton, unlike most of the other octocorals, which are mostly a protein-based skeleton, these have mostly a calcium carbonate-based skeleton. You'll see as we zoom in, there's going to be these long pieces of the skeleton that are white, and these are called internodes, and then they are offset with the protein. It's a little black band, and those are called the nodes. The genus Isidella branches arise from the nodes, and the species Isidella trichotoma, always at the base, it has a 3-pointed branch, or a branch with 3 branches coming out of it. The problem, I'm hesitating now as we're zoomed in. I always wait for the zoom in, don't
Always wait.
Have I not learned that?
Patience.
Yeah. I'm actually thinking this is Jasonisis as we're closer, and that's because of the milky white color of the polyps themselves, the texture. I suspect we're going to see a lot of very small rods. Oh, what's going on there? So if you go back down, you'll see some golden yellow color just to the lower right. Bang, right there. Those are, I think, benthic ctenophores, small benthic ctenophores. Yep, I can see the tentacles coming towards us. I don't recognize this color. So this could be a new species of benthic ctenophore for our observation, I mean. So we've seen lots of ctenophores swimming in the water column. I noted to you, Del, they use the cilia to paddle with, the largest animals that use cilia as a form of locomotion, and there's one group that's become benthic. Yesterday we saw 2 or 3 of those big sort of purpley individuals, and they had those long trailing tentacles with the side branches. I compared them to long line fishing, and this is the same thing, just a much smaller version of it.
Attention all hands. The ship's door will be opening for the next 1-0
Here. Here we have a couple of crinoids, the yellow feather stars, echinoderms related to the urchins that we've been seeing. And then we have brittle stars. They're the kind of peachy orange ones. And these are Ophiuroids, also a kind of echinoderm related to the sea stars and the urchins. So getting some good diversity of echinoderms today. So you can definitely see down below these echinoderms, I can see the branching point where the branch is arising from the nodes. You can see that in a few different places. It's a planar fan, and the texture of those polyps suggests to me this is Jasonisis. We have Les Watling in the chat room from the University of Hawaii. He's also a bamboo coral expert. And he isn't offering a name, but he thinks it's something like what we've seen on the New England seamounts in the North Atlantic. There might actually, right in the center there might be something that is, the skeleton and the tissue is overgrown.
Yeah, there's something up here too by the brittle stars. That
I agree.
I'm
not sure if that's a sponge or an anemone, something interesting going on there. Huh. Yeah, so you can see the very tips of the skeleton. There's no tissue there, and that's where all these things are kind of huddled. And so we have this question a lot. What came first? The associates living on it and have scraped off the tissue, or was tissue scraped off, and that's what is allowing these to settle in this place? That looks like a shell. It must be a hermit crab in there. Looks to me like that's some kind of gastropod shell. Yep, that's what I'm trying to do. So it does look like a shell. The question is, what's inside of the shell? Is it the original molluscan owner, or is it something else that's taken it over? Well, hard to tell. How the heck it got way up here, I'm not sure. Again, a really nice, massive colony. There's a good-sized sea anemone that is grasped onto one of the branches.
How old would you imagine this coral might be?
I hear you saying Chris, so I'm assuming you're asking Chris Kelly.
No, I meant you.
Oh, okay. I thought you were asking Chris Kelly on shore how old it would be. I would say this is easily hundreds of years old, but I don't know how many. The base of the skeleton, it was really, really thick, and you could see a number of different branches arising. I suspect it's put out an initial main stem, and then as it ages and gets bigger, it puts out additional stems, increases the size of the holdfast. There you can see what I mean. 1, 2, 3, 4, 5 at least, and they're really, really thick. base, you can see some hydroids. There's another one of this. That's
yeah.
nice. So 2 of these gastropods. I think that is, you were right, Roland. That's what we saw up above near the top. So some snails. Look like they're both grazing over the hydroids in this particular case. So that's a nice observation. I don't recognize these snails. Haven't seen them before. Another interesting thing you can see here, you're seeing this really thick base, but you're seeing some very small polyps coming out of it. And it's an indication that even though this may be hundreds of years old, the tissue can be renewed, new polyps can be budded. It's an asexually reproducing organism, and so we don't know what the upper limit is to their lifespan.
Yeah, it looks like there's a piece, maybe the end broke off here. Is this a piece of the skeleton down below? Yeah.
Yep. And this is one of the things that people use to do the aging, is they collect the skeleton. They can use isotopes and make a comparison of the change of the isotopes as they are changing from one isotope to another compared to water column, etc., and get some estimate of the age. That was a very poor description of that whole process. Yes, thank you.
Scott, I don't know if you could get to it. There's a colony behind this one that's kind of sticking off the left side of that boulder, and that looks like it could be something different.
I see what you mean, Chris. Stand by. We're just changing pilots, and I will do my best to get it. It looks like maybe that could be a Paragorgiid, perhaps. Let me know when you're settled, Dan. Correct, yep, on the left side. It's got a white stem. Thank you. And I neglected to ask the front row to introduce themselves earlier, and when you feel that is a good time, please take the opportunity.
Yeah, this is Dan Rogers sitting in the pilot seat, and we've got Bobby Moore as navigator, Andy Lister as co-pilot, Roland Bryan, video engineer in the video chair, and Annie White in the highlight clipping chair, and we're all with the Global Foundation for Ocean Exploration.
So indeed, from a distance, this does look like a bubblegum coral, which would be in the family Paragorgiidae. And you can kind of see that the texture feels, I mean, when I look at these, the texture feels a little bit different. The skeleton looks a little bit different. And this belongs to a group of octocorals that are called the Scleraxonia. And the Scleraxonia, the construction of the skeleton is quite different. We looked earlier at one of the yellow sea fans, and you could clearly see this internal skeleton. It was kind of brown, and it was made of protein, and we call that an axial skeleton. You can think of your own skeleton inside your arms, for example. Tissue completely surrounds it, and the skeleton is a distinct unit. In these Scleraxonians, the skeleton is not composed of that material. Instead, the microscopic calcitic elements are all kind of glued together and the tissue's running through it, and so there's no clearly defined skeleton to it. And the coralliids, it's a little bit different. They're so fused together that they're cemented and really strong, so it does seem like it is an axial skeleton, but it's not a protein-based one. So, Chris, what do you think? Paragorgiid or coralliid? I think I'm going to...
No, I totally agree with you, Scott. I think it's Paragorgia for sure. Yeah, just for some of the reasons you mentioned. Also, the very, very clear tentacles and just the general shape sure looks like a bubblegum to me.
Thanks, Chris. And I see 4 astroschumatid, maybe 4 astroschumatid brittle stars. Those are the brittle stars whose arms you can see coiled around different branches. Yeah, let's zoom on the fish. Looks like a rat tail. Understood. Well, he's being a cooperative little fish. Very long dorsal fin, Bruce.
Yeah, so it is a rat tail, a grenadier. This could be either in the genus Nezumia or the genus Ventrofossa. Ventrofossa are often more iridescent than this, but you can see it's quite active. We're still not in an area of high oxygen content in the water, but it's 2 to 3 times higher than it was yesterday when we saw very few fish and they were not active. Long eel-like body, that's very common for deep sea fish because it's a lower energy way of swimming than swimming by flapping the tail like a lot of shallow water fishes do. You can see it's got long fin rays on the fins on the belly, the pelvic fins, and it's got a little whisker on its chin. Sensory organs, but also fairly large eyes, probably for sensing bioluminescence.
No, pilot,
Chris,
we can move on.
do you have any
Thank
thoughts
you.
about the genus?
Astrid thought it might be Nezumia, right? Sorry, Ventrofossa, and I'm actually consulting the guide right now to refresh my memory. That dorsal was really high, and I think that's a distinctive feature that we could pay attention to. Astrid is noting the black dot on the dorsal fin, too.
Yeah, those 2 white colonies are new for the day. The one on the left has some stuff on it, so let's have a look at that one. Thank you. Maybe even a basket star.
So at least in the gallery, Bruce, are the ones that seem to have the higher dorsal fin, if that's important. I just don't know.
So yeah, this is going to be a primnoid octocoral and a little bit of a treat. I think there's a basket star in its arms, and we don't often see those on the corals. Boy, I think I see maybe a metallic gorgia back there. This is already spectacular for the corals. Kind of surprised that it's only 1,027 meters deep at all the diversity that we're seeing here. And there's also a black coral down there. This is a really nice rock. We're going to want to sit here for a little bit, pilot. Great. So this is a primnoid octocoral. This is different from the one that we thought might be a Narella earlier. And I saw one of those Siderid urchins on the backside, and Chris Kelly and Chris Ma had suggested they may be coral predators, and so that may be munching on this coral from the other side. But what we're looking at in the center of the screen here is another kind of brittle star, Ophiroid. This is actually in the same group as those large astroschematids we just saw on the bubblegum coral, except here you can see that the arms are branching. So this is a basket star. I think it might be a youngish one. It doesn't have all that many branches to its arms. So I'm curious about that.
So Scott, I was sort of thinking Paracalyptrophora. What do you think?
You know, I'm always mixed up by the Paracalyptrophora and Calyptrophora, and so I will defer to you, who has looked at a lot more of the images of the primnoids from the Central Pacific here. So that's as good an idea as any that I'm going to come up with you for right now without actually looking at it under a microscope. Yeah.
Well, let's go primnoid because I get confused on calyptrophora and paracalyptrophora as well. So except for I think a lot of paracalyptrophora have downward facing polyps, but I've actually forgotten. I've got to remember whether it's, but that sort of type of branching pattern that you see in Caligorgia, you can also see signs of that in paracalyptrophora. That's why I was going to that.
I did make a cheat sheet some time ago. I just need to find it on my computer here to the primnoids of the Central Pacific, and that might help. So one thing about this basket star, this is a suspension feeder, so it unfurls its arms and it's fishing out of the water column like most of these suspension feeders are doing. They're growing up off the bottom to get into the water flow in order to capture the food. And you can actually see particles moving by in the water column right now, many of which are probably organic based. Let's just have a look at that urchin just down below it and see if we can actually see, because of the side we're on, we're looking at the mouth. Usually we don't see the mouth because it's against the ground, so we can see if the teeth are extended and doing any feeding there. It might be a tough call to see that, but yeah, it's tough. And the other thing is the spines, the way they're oriented, kind of close up around the mouth. What's that on the? There's also some little pink things. Look like, they must be polychaetes, but they almost look like little isopods actually. Yeah, I see that. The one on the middle one on the left, I see has kind of like polychaete legs, but the one that's curled around in front of me, a little bit more mysterious. All right, that's good. Let's kind of, I think there's a whole bunch of zoanthids, those yellow volcano-like structures you saw there. Those are zoanthids, and on the right side, lower right side of this colony, it looks like maybe it's being overgrown. You can see some brighter yellow. Okay, are you able to zoom right into the rock where the laser is now, on the base of the rock? I just want to see all that structure that's coming up from the rock. That's more hydrozoan. So we saw some other hydroid colonies earlier, and that's some more, all that sort of beigey stuff. Thank you. We're finished with this one. That's good. No, that's another version of the same colony. We can look at the little stem in the foreground of that white colony on the right. It's kind of brown. Yep, brown pink. That's a black coral. That may be heteropathes. Yesterday we collected a heteropathes. Go ahead, whoever's on shore. No, it's a
all I
bathypathes.
was going to say, Scott, since you're pointing this out, is I think there's also a Metallogorgia behind one of these colonies as well. I got a glimpse of it earlier.
Yeah, I saw that when we were settling down, Chris, and I noted something, and that's when I said I'm really surprised at 1,026 meters at some of the species we're seeing. I would expect to see them a bit deeper and not this shallow. So I do believe this is a hexapathes. We collected one yesterday, but this one has two chirostylid squat lobsters in its branches. Thank you, pilot. That's great. What a great rock here. So now these colonies, notice that these yellow colonies on the right, they're slightly different yellow color, and they do look like they have ophiuroids in their branches, which would suggest those are plexaurids, and those ones that are more deeper gold color, those are the acanthogorgiids. Absolutely, if we could. Yeah, so I see the direction you're coming around. Okay, if we come on the right, yeah, let's get a look at both the yellowfin and then I think it's a Metallogorgia, but we want to find out if it's that or a Chrysogorgia. So that little tuft of pink that's sticking up above the rock behind the yellowfins. Copy that video. And there's also another kind of Chrysogorgiid, I think. Maybe it's a primnoid growing off the corner of that rock on the right. So this must be a Metallogorgia because I can already see at the center of it the brittle star that is always in the center of these Metallogorgia colonies. I want to see the yellow fans in the foreground just by the laser first.
Yeah, there you go.
So they're definitely different from the acanthogorgids. It's some kind of plexaurid. I won't go as far as to say that it's a Paramaricia, but it's definitely a different species. So it's kind of nice. The two things help me. The slightly different yellow color, but seeing that large brittle star. We didn't see it in any of those other colonies. So it's a really good indication that there's a close host relationship and associate relationship. The brittle star isn't just settling on any species of coral. There is a specificity to it.
Yeah, we saw dozens of those other corals without a star. So.
Exactly. So I suppose this could be some kind of Paramarisid or Anthomericia or something like that. And I think there's a little bamboo coral trying to grow out from behind it. All right, thank you, pilot. And let's go to the far right of the rock on the edge here. I know that's Metallogorgia in the center. Yeah, oh, okay, it's a primnoid. We're good. We're good on this rock. Very nice. Beautiful view of that sedared urchin from this side, and another basket star on the left.
Yeah, I didn't notice
colony.
that from the front.
Yeah. So two basket stars, both on the possible Calyptophora colonies. So much looking at the screen, I'm missing a bunch of comments from our scientists on shore. Look like they're talking about some of the fish and identifying the rat tails. We can have a look at the white colony in the foreground. The lasers are passing over it right now. It's a little feather, a whitish feather. In the background, it looks like there's a bamboo coral that's been completely stripped of its tissue. You can see the alternating dark nodes and white internodes. This is going to be a black coral right here. And parantipathes, and at least in the Atlantic, whenever we see these, we see two chirostylid squat lobsters really small in the branches. And I notice there's another one in the back right that's branched. That might be a different species as well. So indeed, a black coral. No, not so. Yeah, it could be Parantipathes, the genus Parantipathes. And I'm looking to see if there are any little squat lobsters in there. Looks like there's some copepods, and there might be little worms, those orange things. You can see very tiny, delicate polyps sitting on top of very short, what are called pinnules. These branches in the black corals are called pinnules. But I'm not seeing any of the squat lobsters. That's a good view for us, thank you. All right. Okay. As you turn to your right, or as you continue to go right, there's going to be another, I think it had two branches, same sort of color. There it is. It just came into view on the right. It's to the left of that whitish bamboo coral. And I think this is another kind of black coral. And what'll be interesting to see is if the pinnule structure, yeah, I see that crab. We'll do that. Whichever one's easier for you to do first. Okay. Oh, great. So this looks like one of the carrier crabs or porter crabs that is carrying an anemone using its fifth pair of legs over the back. And we've seen these carrying bits of octocoral, bits of black coral, some zoanthids. And this one's carrying an anemone, and the idea is that it's serving as some protection, kind of hold it up above itself. So just holding it with a reduced pair of legs at the back there. It's not actually stuck to the carapace.
Oh,
Thank you.
I see. Huh.
Good view. There are other kinds of crabs that literally do glue it to the carapace, but not that group.
So would the crab of sought this anemone out then?
It's entirely
Is that
possible,
possible?
sure. So this is a new fish. Is Bruce still there, or Astrid?
Yep, I'm here. This is a duckbill eel in the family Netostomatidae. Not quite sure what we're looking at here. It's rather elongate. There's a genus Venefica that's got a proboscis on the upper, on the snout, basically. And this one looks like it has a short proboscis. This one's very elongate and pale colored compared to some that we've seen in the Hawaiian archipelago. So it could be a different species. But there are a couple of other genera, too. There's one called Netostoma that's a bit different than this. It doesn't have that proboscis on the snout, and it's kind of a stockier-bodied duckbill eel. There's also a poorly known genus that I'm not very familiar with called Nedenkelles, and I'm not sure if this could be that. This could be a Benefica with a short proboscis. Benefica comes from the Latin word for sorceress. So these are called sorceress or sorcerer eels.
Terrific.
In addition to being called duck-billed eels.
If we could, yeah, this fish, and then above it I think is a sponge. Since the fish took us over here, why not have a look at that sponge, maybe the first of the day. So Bruce, maybe another rat tail. This is different, doesn't have as big a dorsal fin as the last one we looked at.
Yeah, I think this one is probably one of the Nezumia species, definitely. We talked about that as a possibility for the last one that we saw. There are a number of Nezumia species with different morphologies, and probably more down in this area than we know about, because most of the species from the Central Pacific were either described from the Hawaiian archipelago, the Nazca and Sally Gomez ridges to the east over near Chile, or New Caledonia. So you can tell that those locations kind of form a broad triangle around the Line Islands, and those points are far distant. But my guess is that this is a different Nezumia species.
Thank you, Chris. And I also noticed it was going over something on the rock. It was a really small brown plate-like. That was a foraminifera, xenophyophora, I think. Yeah, on the upper right, there's a frilly-looking sponge, whitish gray in color. May even have an ophiuroid on it, because I see a pink spot. Unfortunately, Chris Kelly has gone to lunch. I think this might be a Hydroid, and I am rapidly trying to find where I put that information. Let's see, here we go, Hydroidae. So this does look to me like it may be a sponge in the family Hydroidae. This would be a kind of glass sponge. Formal name for that would be the Hexactinellida. And glass sponges are so named, A, because they have spicules, these microscopic calcareous elements, not calcareous, microscopic elements made of silica, glass. But most sponges have silica spicules, but in these ones they are long and have 6 rays. So yeah, you see this kind of interesting frilly appearance. Sponges, like the corals, are home to many other organisms that'll climb up and on it, and in this case you can see inside of it. And I can see a crab. Let's see. So this is a Galatheid crab, so that would be classified as a squat lobster. Not sure what kind, maybe a Munida type, not the chirostylid. Little tiny chrysogorgid there down on the rock, but I'll focus back on the sponge. And the one inside, I'm trying to tell if that is a snail or a hermit crab. I can't tell if I'm looking at an antenna, a crustacean antenna, or something else. That's good. We're happy when video's happy. So a Hydroid sponge. It's our first glass sponge of the day. There's a black coral back there. I mean, there's so much to look at. We could spend our entire dive right here. It's amazing. It's amazing. Well, I was just referring to this white whip that's at the bottom center, slightly right of center. It's a bamboo coral, and maybe we can just get a look at the polyps, help with some level of identification.
Oh.
So bamboo coral again, we can see the white internodes with the dark nodes around it, and what I'm looking for right now are two things, some details of the polyp, but also the distance between those brown nodes. Do they seem fairly long or do they seem fairly short? And to me, they seem fairly long. So this might be a very young colony in a group we call longbones. Oh, look at that, the end of the skeleton, that's great. So it actually has a developing polyp right on the tip. So that is also an informative thing. It's kind of like the leading edge.
Right.
You could see there were some sclerites around it. I'm sorry, did someone call me? No, I'm just hearing voices. Cool. No, that's okay. I got to see it. Basically, what I could see is that it wasn't being led by just the point of the skeleton and tissue. It was actually being led by a polyp itself. So that was that's good. No, we can move up. Most of the tissue is gone, and as we passed over with the fish, I saw some polyps. So that's okay. Thanks. There's plenty to see.
So as the skeleton grows, what happens to that polyp on the end? Does it shift to the side? Does it move up with the skeleton?
What a great question. And so, you know, the polyp itself and the tissue around it, that's what's depositing the skeleton, right?
Right.
And we could look on some of the bigger ones and see what's happening at the apex. Is there a polyp there? Or does it, as you say, kind of turn and then the skeleton goes up from it? And we don't know. We would need to come back and do a time series analysis. And it's a really good point. You know, we said many times this is a expedition and voyage of exploration. We make these observations and then we come up with these great questions, just like you asked. And what we would have to do is come back and be able to monitor it for some period of time here in the deep sea. Of course, we can make comparisons to shallow water corals and how they grow. The problem is with this group of bamboo corals we're looking at, they're restricted to the deep sea. You don't find them in shallow water.
Okay.
And so we can only answer those questions from looking at deep sea forms. Current moving southwest to northeast, and it's a little bit stronger than when we first settled down. Thank you, pilot. So again, here's a colony. You can see the hold fast. I think it doesn't look like it's alive. Probably a bamboo coral. Yep, it's a bamboo coral. And the rock seems to be tipped over.
Yeah.
So they're not all in place.
No.
Do you want to get a rock?
Yeah, well, if we can get a good thick one. What we're seeing here are lots of these little thin plates here.
Well, I think I see a rock that might be loose.
You want to grab that one?
It's got a big old coral on it.
That would be a perfect one.
Well, we were just half joking about getting a rock sample, and what I was noting is that this coral appears to have grown and then tipped over, which suggests that the rock is loose. So I was suggesting that was a fine rock. But in all seriousness, let's just see if it's alive, this colony.
Yes.
It is alive, yeah. I can see it's alive at the tips, but it's being fed upon by a sea star. So maybe we should look at the fish first, and then we'll go back and look at the sea star. That'll give Chris Ma some time to call in ahead of time. And I suspect it's a Hipposterid corallivorous fish, sea star, sorry. Bruce, would you like to talk about this eel-like fish? There's one of those.
Well, this is a cutthroat eel in the family Synaphobranchidae. I'm not going to offer an identification right now because Ken Sulak is on the chat room, and he is a world expert on this group. He revised the genera that this is a member of. I think it's in the genus Synaphobranchus. they're called cutthroat eels because they have the gill openings under the throat next to each other, like somebody's put a slash across the throat. I see that the dorsal and anal fins are about opposite each other. Anal fin origin is a little bit further back than the dorsal fin origin. This, my tendency would be to call this Synaphobranchus affinus, but when we get a reply in the chat room from Ken, he can tell us definitely what this is.
All right, thank you. And I just want to note that fish right now is curling around one of these xenophyophores that I pointed out earlier. So that's a structure that's built by a single-celled organism. And as we're following the fish, I heard a beep, and I suspect, Chris, you may have joined us? Wonderful.
I am here.
That's great. So we're now just going into a zoom on this bamboo coral, much of which has been stripped of its tissue on the lower part of the colony. But we can see on the upper outer edges there's still some tissue, and I see what I believe is a hipposteriid urchin. But I think I'm also seeing, believe it or not, an echinotheriid urchin on another branch. So we'll have to check that out as well. But have at it, Chris.
Just waiting for it to come a little closer. It's definitely a goniasterid, however. A lot of goniasterids we're discovering feed on cnidarians, such as these corals. It's not entirely clear to me if it's a species-specific thing or if it's something that is just a generalized type of predation. The more goniasterids we see that are feeding on these, the more the pattern sort of has to adjust to accommodate what we're observing. That is an echinotheriid on the branches, isn't it?
Yeah, isn't that weird? We'll look at that after the sea star.
That is weird.
Yeah, glad you agree. I'm wondering if the echinotheriid is just kind of cleaning up the bare skeleton. If there's, bacteria or something growing on the bare skeleton, it's kind of munching on that.
Yeah, I mean, I've also wondered whether or not a lot of these animals are simply just taking opportunities, because of, free meal. Maybe with the coral downed, maybe whatever defensive mechanisms it normally has in place.
Sure.
Not able to repel the urchin during a normal situation. Before I forget, so the sea star definitely looks to be a hippasterine type goniasterid. The spines on the surface certainly suggest that. I think it might be evoplasoma. Usually when we see hipposteria itself, the spines are much larger, and the body form is a little more, the arms are a lot plumper. It's one of these things you talked earlier about how these are essentially a series of pilot studies. Observations give us ideas for coming back and studying these in greater detail. And one of the things that, if we were to study these in a more elaborate context, I think one of the things that would be really interesting would be to come down and look at how many of these, are seen, how frequently, but also how long, these occupy these coral prey. As you, observed years ago, which I was happily able to publish, there was a goniasterid circiaster which ended up on a paragorgia and was feeding on it for almost a year, which was just remarkable.
Amazing. I don't see the stomach out on this one.
The echinotheriid is. I'm sorry, go on.
I was just going to say we've got a zoom. You'll see it in a second of the possible evoplasoma, and I don't see the stomach out. I can see the tube feet hanging onto the branches, but doesn't appear to be actively feeding right now.
Yeah. Well, I mean, I think, like you said, one gets the impression that essentially when they found a big food source, they just stop and park themselves there, and then they kind of stop and eat whenever they want. I mean, it's like when I was watching, I was at the movie theater the other day. You like to stop and you don't like to go and buy extra popcorn. You just like to have a big one. And every once in a while you stop for an hour and then, then when you're hungry again, you start on the popcorn,
There
and
you
then
go.
when you're done with that, you go to the chocolate raisins.
Good,
So
good
this
comparison.
coral is the chocolate raisins and popcorn all mixed up in one.
I'm looking at the branches of the bamboo coral as we're zoomed in here, and you can clearly see the internodes and nodes. And this has internodal branching, not nodal branching. The branches are coming out just after the nodes. And I'm going to guess with those the polyps that are retracted, that this may be maybe D2 clade. You can see some of them are extended right here, but if it is that clade, I'm hesitating. So if it's that clade, there should be some rather prominent needles. I can see needles between the tentacles, but they're kind of short. That's good, pilot. Let's go down to that urchin on the lower left. Thank you. Right, so you can see now this one has been overgrown, this part of the skeleton, with hydrozoans. And what's kind of interesting is most of them appear to be growing on the protein part of the skeleton and not the internodes. So that suggests to me that the larvae, when they're swimming around, preferentially choose the protein and not the calcium carbonate base. That's really interesting. So Chris, what do you think about the urchin?
Well, it's an echinotherid, that much I can tell. It's interesting because the test pattern certainly suggests it's an echinotherioid type urchin, but I noticed that there are no little horseshoes on the spines. So it might be something a little different. I'll have to confer with some of my colleagues.
Can see it.
This is a very difficult group of animals to work in, just because the skeletal patterns are destroyed so easily by collection with trawl nets and so forth. But this is fascinating. I mean, I've heard of them feeding on kelp and other vegetative matter in the Bahamas, but this is the first time I've ever heard or seen them feeding on, or I'm assuming that's what they're doing, is feeding essentially on these hydroids or whatever other kind of organic material is presumably on this bamboo coral.
Yep,
so
I agree
I'd
with
say
you.
this is a really novel observation. That's fantastic.
Well,
I don't know what it's doing on there. So
I just love seeing all those hydroids restricted to the nodes. That's fascinating as well. That tells us a lot about settlement. And I was going to say, you'll see the imagery later, of course, when you review the other images in the video, but on that close-up, you got really good views of the pedicularia on the end of their stalks on that urchin.
Oh.
So that'll be cool. And just one final observation, Chris, hang on a second. I just want to point out how big the holdfast is for this colony on that rock. Another good indication, that's the part that's attaching the colony to the rock. Another good indication that this is probably a very, very old colony, and it's become very stable attached to that rock. And I have to believe that that has shifted somewhat during its growth, but maybe not.
And
that's
I was
good.
actually going to follow up with it. Oh, yeah, I'm sorry. I was just going to follow up with what you just said, because it looks like it's fallen over somewhat. And I'm sure that that's probably part of the reason why both the urchin and the sea star have suddenly taken advantage of it. I mean, it's a lot easier to reach from the bottom, in terms of physically climbing up onto it. So, if I was to look at the succession, I'd look at the sea star first, and then that urchin probably crawling up onto the animal, onto the coral afterwards. It being, I've seen other instances of, I guess what you call opportunistic predation, where I think sea stars that we wouldn't normally associate with coral predation just seem to be crawling over, falling over corals, and you kind of walk into a crime scene, you don't know what's been happening, and potentially that is because you see these predators on these corals, and sometimes you make the assumption that that's what they're always feeding on, but sometimes I don't know if that's always the case. The hippasteroids are definitely the case, but like those urchins, the cineroid urchins, I could see feeding on them, but the echinotherid might be there just opportunistically. So, but who knows? We'll see. That's that sort of peppermint-colored one that we've been seeing, and it's not like the purple ones that we've been seeing. So, different species.
To
wow,
go,
that's just great.
yeah. To go back to your popcorn
Anyway, I'll let you get
chocolate
back to
raisin
it.
analogy, I would say that
Sorry,
it's. To go back to your popcorn chocolate raisin analogy, I would say that it's easier to reach the cookies on the counter than it is on the top shelf. So when they tip over, they're easy pickings.
Splendid. Oh, yes.
Thanks, Chris.
So good luck, and I'll talk to you in a little while later. Thanks.
So this is a really interesting observation. We're seeing a hydrozoan coral, and so really interesting convergent morphology. And hopefully as I'm talking, I'm going to remember the name. I'm having a moment here where I can't remember the name of the genus of this Solandaria. There we go. So this is, I believe, Solandaria, and I think there's a sea spider under there. Is that what's going on the left side? So you see it looks like a sea fan, looks like an octocoral, but it's not. It's actually in a different group, the Hydrozoa. And we've seen these giant solitary hydrozoans.
Yeah.
We've seen some very fine, small hydrozoans. No, that's just a shrimp. My bad. I don't mean just a shrimp, pardon me. I mean, it's not a sea spider. It is a thread-legged shrimp. Thank you.
It's okay. Shrimp day's over. So shrimp
That's
had
right.
its
That
time.
was last week, right? Yep. Yes, Nav. Yeah, so really good question, Nav, and just in case not everyone's hearing that question, it's basically why is it that these corals may be tipping over? Is it because of weight? Is it because of current? Something like that. So it's more the current. I've seen some studies of some black corals in fjords off of British Columbia, really interesting. They settle on fairly small rocks up near the sill of these fjords, and so there's water that's kind of moving tidally in and out. And they grow up as a fan, and they're growing that way in order to intercept the maximum current flow. And when they get to a certain size, the current is strong enough that it pushes them over. So they're laying horizontally, but they're still alive. So they curl up, and the branches regrow in a new orientation so they get back into that flow. But then the current catches them, and it drags the whole rock over the sill and down into a deeper depth where they can't feed as well. So I suspect here that's what's going on. They got basically too big for the britches. The base wasn't strong enough and fell over. But if it stops in a certain position, then they can continue to grow. The white colony may be something new.
Another fish on the left. It
Yep.
looks like another rat tail or something like it. It could be. This one looks like it's in a different family, actually. It's a related family called the Bathygattidae. And you see the long extensions on the dorsal fin, on the pectoral fins, and the pelvic fins. This would be in the genus Gadomus. We're not sure what species because this family is badly in need of a taxonomic revision. So this is a relative of the rat tails. I don't know if they have a common name, but genus Gadomus, and they're quite graceful. You see the long whisker on the chin, the long thread-like rays on the fins on the side behind the head, on the belly, and the one on the back. This one has some beautiful iridescence on the head that I haven't seen before, reflective scales. So a Bathygattid in the genus Gadomus.
Yeah, so if we could have a look at the white colony to the left of the lasers, please, unless there's another fish.
There's starfish lower left of lasers, straight down from lasers now.
Okay, I want to check out that coral first. I think this is another, yep. All right, big fish coming up on the right ahead. Big fish shows us that there's another bubblegum coral over there. Wow, look at all the coral. It's behind you, behind and above. Oh, sorry, this is a replay I'm looking at.
Okay.
Understood.
Yeah, he's messing with you.
Don't move. Turn to the right, please. And there's a whip coil. Oh, there it is. There it is. That's a shark.
Wow.
Run, Bruce. Looks like some claspers there.
So my guess is that this is a small-tooth sand tiger shark. You can see it's got some impressive teeth. It looks like it's a little different shape. 5 gill openings on the side, which most sharks have. That eliminates 6 gill shark. So I think it's Odontasmus ferox. It looks like it's got a damaged mouth.
Yeah,
like it's somehow been hooked and damaged or something. It is a male because of the claspers on the pelvic fins, the back toward the back. I'm quite open to correction on the identification of the small-tooth sand tiger shark. They're not in the same family as the tiger sharks in shallow water, different family. This is a deeper water one. Very impressive.
I agree. Yeah, for a change, this is one that we're not chasing away. You can see on camera too the view of the D2 ROV with this shark to get a sense of the size.
Yeah, well, at least a couple of meters there, huh?
There's also another fish down just below the shark to the left.
Thanks, Astrid. We did see that. We're just, the vehicles have got to get back into position with one another after chasing the fish. Or the shark, excuse me. It's not just a fish.
Shark's a fish?
Jen Cassell, who's in the chat room, she's at UC Santa Barbara, notes that she believes this is also, she also believes, excuse me, this is a small-tooth sand tiger shark. And she notes that they act like this with subs and ROVs. Very curious. Copy that, pilot, understood. So we'll just be a couple of moments here while we deal with a loop and a tether from our little excursions here. So stand by. We're about 6 meters above the bottom right now, and it'll take just a couple of minutes to deal with the engineering operations here. Ken Sulak from the USGS has made an interesting observation with respect to that sand tiger shark that we were just looking at, and we noted that on the left side the mouth appeared to be damaged. And he says that in the Gulf of Mexico at about the same depth we have repeatedly seen the same individual shark recognized by a damaged caudal fin on separate cruises that suggest that sand tigers are resident in a small area. So perhaps that shark was patrolling its home area. Thanks, Ken. Very interesting observation. Jen Cassell follows up that a small sand-toothed tiger came blasting out of the murk when she was in a sub in the Desventuradas Islands off Chile, and it attacked their sub. And Ken Sulek says he has had the same experience with hammerheads on 3 Johnson Sea Link dives. Shark zoomed right in and banged into the upper work bar above the sphere where lights, sonar, and other electronics were affixed.
It's a little too curious for my liking.
Yeah, that's what I noted when it was pointed right at us. It's like, glad I'm sitting up here and not down there.
It's one of the advantages of ROV work here.
Exactly.
Yeah, so if you're just tuning in, you're watching a live video from NOAA's ship, the Okeanos Explorer, and we're about 360 miles north of the equator today at about 162 degrees longitude. And we're just paused here for a little bit while the ROV pilots sort out a little twist in the tether line. We were chasing a shark just a few moments ago and got things in a little bit of a twist, but we'll be sorted out here soon and on our way. This is our third dive in the Kingman Palmyra area, and part of our effort to explore the Pacific Remote Islands Marine National Monument here. Today we're on the southeast slope of Kingman Reef, diving currently at a depth of about 1,000 meters and trying to work our way up to about 850 on the summit of a small cone on the slope of this larger reef seamount complex. Kingman and Palmyra Reefs are really the kind of exposed summits of just 2 seamounts in a much larger chain known as the Line Islands here in the Central Pacific Ocean. We think that most of the volcanism here happened a little bit around 90 million years ago, and then another pulse around 70 million years ago. And at this time, these seamounts and these islands would have been building on ocean floor that was already probably 30 to 50 million years old. These islands are kind of the last sub-aerial remnants of what was a much larger volcanic platform, about 200 kilometers in diameter. If you go into Google Earth and look at it, you can see that it has sort of a horseshoe shape to it. And this elevated platform has subsided as the seafloor aged. We can see, if you look around regionally, that these seamount tops that are above about 1,300 meters tend to have flat tops, we call guyots. It's a sign that they were once at or near the surface and were cut by wave action and may have had reef material built on top of them. And then the larger seamounts that are deeper than about 1,300 meters tend to have more pointed or conical peaks. Interestingly, we're on a platform here today where we're diving on a fairly small cone on top of this larger platform. So one of our questions here is, what is the kind of the nature of the rocks? So far, we've been seeing is lots of manganese crust over them. We've been seeing lots of bigger blocks, though. This might be an indication that I think some of this is carbonate material, but it can be really challenging to see what the rocks are when they're covered with this thick manganese crust that we typically see on these older rocks here in this part of the world. As we've built up a large area of volcanism here through time, we've basically thickened our crust in this area. So folks who look at gravitational attraction here in this part of the world, they measure gravity. From those data, we can sort of tell how thick the crust is, and there's been an indication that The crust here is thick enough that it's more or less, we would say, isostatically compensated. I mean, it's sort of floating. That low-density crust is floating in a denser mantle, and that may actually have kept these islands in this carbonate platform a little higher than usual. It's quite a bit higher than the surrounding abyssal seafloor. It's still subsiding slowly, but maybe there's a little bit of buoyancy effect associated with having that thicker crust in this area. As we've been moving north here, one of the things we've been noticing in the larger scale topography is that we have a lot of submarine channels. In fact, we can see that the carbonate material is coming from these islands and actually moving out into the deeper slopes. But on top of this volcanic complex, these reef materials have grown through time, and they've been growing at a shallow enough depth where the rate of carbonate accumulation from these corals, shallow corals, has been greater than the rate of dissolution. So we're able to build up this source of sediment, which is now working its way into deeper water. So we come up now about 30 meters off bottom. So if you're watching D2, you're sort of seeing the lasers going out there into the water column. And again, we're just trying to sort out a little tangle here on the tether. And once we have that worked out, we'll be back underway. So stay tuned. You can sort of see what's going on. I think on camera 2, that's the view from Sirius, the upper ROV. And it's that tether that connects Sirius to D2 or Deep Discoverer ROV that's got a little twist in it. And of course, we want to sort this out. We don't want to, if D2 should pull away from Sirius and put tension on that line, we could damage that cable. And that's what the ROV pilots are working on right now. So again, if you're just tuning in here, we're RV pilots are just starting out a little kink in the tether before we move forward. But we're diving today in the off Kingman Reef in Pacific Remote Islands Marine National Monument. And the ship has been out here doing work as part of the Capstone project, a campaign to address Pacific monument science, technology, and ocean needs. And this has been a multi-year effort that NOAA has been involved in, really to explore the deep water areas here in some of these marine protected areas, such as where we are today. So there's been some observational work done in shallow water here, but not much, or in many cases here, not any work done in deeper water to look at the geology and benthic and pelagic habitats. So this is the work we're out here doing. We should mention that all the data we collect in these expeditions is made publicly available, and so you can anyone can access it, scientists from around the world, the general public, and that includes our sonar mapping that we do from the ship, as well as the video and these information about the samples that we're collecting here with the remotely operated vehicle, or ROV. So if you're watching on camera 2, you can get a look at this little kink that got developed in the tether as we were sort of chasing a shark. And of course the pilots want to make sure we take care of this because we don't want to pull on that kink and damage the cable in any way. So one of the challenges, we're working today at about 1,000 meters deep, and so one of the small challenges that arise here from time to time, but we're hope to be back on track soon. If you're watching camera 2, then what you're looking at is the Deep Discoverer vehicle, and then you can see this yellow cable which comes and connects to the Serios vehicle, and Serios sort of hangs more or less down from the ship, and then this tether connects it to Deep Discoverer D2, And it's meant to be buoyant like this, so that when Sirius moves up and down with the ship, of course, it takes some of the heave out of the bottom ROV D2. And you'll see the yellow cable there, the buoyant cable, and you'll see two other thinner lines. One of them is a recovery line we use when we bring the vehicle back up to the surface, and then there's another line that sort of holds that recovery line onto the yellow tether. And so as we're maneuvering today, we got this small kink here, and so just to give you a sense of what you're looking at with those various cables in the view. Okay. Great. Yeah, so everyone out there on shore, we're back online now. We're ready to move again. And we're going to have to be a little more conservative with some of our movements because of the tether situation. So please keep that in mind when you make your request. And if we see a loose rock right here, we could go ahead and try to grab it, I guess. Looks like there were some up forward here that might have been loose. Yeah, if we could while we're sort of setting up here, if you see something, we can. The bigger, the better. So looks like some of this may be loose. So. Yeah. Can you get the one right below the lasers there? Yeah. Curious if it's. I'd like to get something kind of thick. I can't quite tell if that's kind of tabular or not. So maybe we can. Okay. Yeah. Let's see if that's loose. So I guess we can. All right. Yeah. Feel free to give it a good squeeze. All right. Yeah. Looks like a pretty good chunk there. Get a great. Looks all right to me. I think we got a few associates to keep Scott busy for an hour or two after recovery. All right, great. Thank you, pilot.
So the little fish is another bathygatid, one of these relatives of the rat tail that has the long fin rays on the dorsal, pectoral, and pelvic fins, and the long whip-like tail.
Great, thanks, Bruce. All right, so right now we're at a depth of 1,022 meters, it looks like. We may have come a little further back down the slope when we reacquired bottom. So we're going to keep working our way up this small cone again on the edge of Kingman Reef.
So Del, what would cause all these rocks to be broken up like this?
Yeah, so it looks like many of these rocks are out of place, and sort of that, they look kind of like tablet shape to some of these. This looks a lot like it may actually be carbonate rubble coming from the reef. I assume at this shallow depth, we might have had some carbonate material develop even on this little cone, even though it's not very flat at the top. But it can be really hard to tell what the rocks are when they all have this thick crust on them, which is a little frustrating. So one of the things we'll be able to do if we get this rock up is actually see what's inside of it. If it's volcanic rock, then we can hopefully look at its geochemistry and get an age date on it. That's why we try to collect biggest rocks that we can, because through time, of course, seawater will diffuse into the rock and altering its chemistry. So we hope that there's some unaltered piece there in the center. But yeah, so if it's volcanic, we try to do that. Otherwise, at least we'll have a sample of it or know for sure what was exposed down here, what type of rock when we get it up. And then in this case, it had all sorts of little things living on it, so it serves a dual purpose. I think we've seen all this here. We can move. I think we can go ahead and move up slope here. And Scott's just back from lunch, so yeah, we did get a rock sample while you were gone, and we just sort of did that as soon as we sat down. So we're ready to go ahead and move. And
excellent.
I think we've got a few people back online. Bruce was describing a fish we saw, so hopefully you're not. People have come back on now.
Great, good to hear. Yeah, I think we should try to get some way up slope. We saw a lot of different kinds of corals and diversity down here, and want to see if we can make our way towards the top of the cone. Okay. Go ahead, Nav. Sorry, Nav, I'm here now.
So this is another one of our holosaurs, the spiny eel relative. You can see it's like a true eel, but it's got the short fin on the back that the true eels don't have. We're looking at its belly right now, but I suspect it's another Aldrovandia phylacra, but I could be wrong about that. It looks like it's got a shorter head.
Pilot, can we get a zoom on this white colony in front of us, please? Thank you, Nav. So we're looking at a black coral close up there, and that might be the genus Trissopathes. We're just getting an update on the ROV system status, preparation for the remainder of our dive here. And just stand by and enjoy the scene as we're having a pilot change, shift change. see this area is rich in these yellow sea fans, and the close looks that we've had before suggest that there are a couple of different species. Some of them are plexaurid sea fans in the family Plexauridae, and some of them are Acanthogorgiidae. Good afternoon, Josh. It has been a very exciting morning on various fronts, and as you can see, there's a lot of coral down here. We've been having a pretty good look at a lot of the stuff that we've seen down here, and we're kind of overwhelmed with the diversity, but we would like to make some move upslope. So I think that's our challenge now. Wow, that's great. 4 hours of bottom time. I'm going to like these shallow dives. Shallow-ish dives. Thank you, Nev. This is what we refer to as a Venus flytrap anemone, and it is come to rest on what looks like a bamboo coral skeleton, long dead. It's been overgrown by a variety of hydrozoans. You can see a curled up brittle star there at the base. But this, you see this orientation of the oral disc very frequently in these particular anemones, and presumably it's a better way to capture zooplankton and other creatures and food bits that might be going by it. Thank you, pilot. We see another, the purple coral down there right at the base where you zoomed in. That's new for the day. And we believe that may be the genus Victigorgia. It's very deep purple. It's interesting. I don't recall, maybe Chris Kelly or someone else, if they're on shore, or if Tim Shank is on shore, can remind me if we typically see the astroskematid brittle stars in those branches. I don't remember that. Doesn't look like Tim is with us today. I see Sonia Rowley is in the chat room. Maybe Sonia, you can remind me if you've seen many of these before. But it's just a beautiful, striking purple color. And I believe this is another congrid eel.
No, this is actually another one of the cutthroat eels, one of the Synaphobranchus species that we saw before. I didn't get a quick enough look. I was talking to a couple of visitors here, but so I didn't get a look to see where the origin of the dorsal fin was with respect to the anal fin, to see if it was the same species that we saw before. If Ken Sulak caught a glimpse of it, he could tell us what it was. So we'll wait and see what he says. But it's another Synaphobranchus.
Thanks. In my defense, when I said congrid eel, I did mean to say Synaphobranchus.
I had a hard time seeing the dorsal as well, so I think it was kind of hard to see it because it was swimming behind that coral.
Looks like we're passing over a dead sponge there. One of the things I was saying is we have two kinds of yellow fans here. One are Acanthogorgiid fans and one are Paramyriciid fans. And one of the ways you can tell them apart is the Paramyriciids always seem to have a fairly large brittle star in their arms, and the Acanthogorgiids don't. And this looks to be, I'm guessing that this is a Metallogorgia skeleton that has become overgrown with hydrozoans. So the really thin, lacy stuff that you're seeing, that's live, and in places I can see very small tentacles sticking out, so those would be hydrozoan tentacles. But the skeleton itself has that dark, iridescent color that suggests it's a Metallogorgia, and we did see a few Metallogorgia colonies alive earlier. Thank you. This is kind of a leggy looking coral here, not kind of a full fan. I'm not sure why that is. Presumably as it's growing and putting out its branches, it's responding to the currents at the time that the branches are being formed. So this may inform us something about the local conditions at the time of branching. You can see a lot of brittle stars on this colony. And there was a black coral just to the left of it, I think a Trisopathes genus. Work that we've done in the North Atlantic canyons at approximately the same depth. These yellow paramyrcid sea fens are also extremely abundant and common there. So this is a really interesting observation. Different oceans, and yet the same general morphology, color, and possibly the same genus. And the other thing you can see is, at least for the yellow sea fens, they all seem to be oriented parallel to one another and perpendicular to the mean flow, so the average flow, which must be coming back and forth across us. And earlier on, the ROV pilots did note that the currents were moving from the southwest to the northeast. Bruce looks like another, or Bruce and Astrid looks like another rat tail here.
You know, this is the same one that we saw earlier that has the sort of bulbous head with a concave forehead. And we were debating if it's a Nozumia species or something else. There's another genus, Nozumia, that has a large rounded head. I suppose it could be that. But for the identification of these rat tails, it's pretty much guesswork for us because we don't have any of the experts online. And there are dozens of genera and maybe a couple of hundred species in this family, so they're rather difficult to identify. Some of the first characters are things like presence or absence of a light organ on the belly and the position of the anus relative to the anal fin, things of that nature. So it could be Nozumia or a similar genus.
And pilot, if we could hold here just a second, the two corals that were there, just to make the note, so the yellow one on the left, that's what we've been saying is Paramyrcia. That's classic with that large single brittle star in its branches in the center of the fan. And then to the right, a completely different family of coral, the Primnoidae, but it also has an Ophiuroid brittle star in its branches. And just as Bruce was saying that it's really difficult to distinguish some of the characteristics of the fish, it's equally difficult for us to distinguish the characteristics in the video of the brittle stars. So one thing we don't know from this view, we know that both of these very different kinds of corals have these brittle stars. And the question is, are they the same species or different species of brittle star? That is, does one species of brittle star prefer one species of coral? Thank you, pilot. This is some of the work that Tim Shanketwood's-Hole has been doing, and you've probably heard Tim speak several times. We've been tuning in over the last couple of weeks, and he's interested in this relationship of the associate and how tight it is with its host. Kind of a large vase sponge just off to the left. Yeah, we've seen the rock pen several times, but I said left, I meant right. I always do that. And I think it's dead. There we go. Yeah, so it's a dead sponge skeleton, glass sponge. I suspect a euplectellid of some kind. But we haven't seen any like that alive so far today. I don't know if Chris is still on the line.
Yeah, so Scott, I think you nailed it. It's a dead euplectellid vase. My guess would be it's either Dictyalis or Regadrella, probably Regadrella, but not sure at all. Anyway, one of the things about these dead sponges, and not that I'm advocating a collection because I'm not, definitely not, is that when they do die, they lose a lot of their microscopic parts, the spicules, the very, very small ones that are embedded in the tissue, and therefore they don't. In order to really nail down the taxonomy of a sponge, you've actually got to collect a live one. So anyway, we can just tell from the morphology that it's in that family and probably genus Rigodrella.
Thank you, Chris. We've been seeing these black coral, they're seemingly unbranched, but they're actually branched. They're kind of like little test tube brushes. Probably the genus Trisopathes. Tina Molitsova from the Shirshov Institute in Russia, who's an expert in the black corals and been helping us extensively, is traveling. She's on her way to join a cruise of her own, I think down in Tasmania. Looks like we're going over another Metallogorgia colony. So just a few moments ago, we imaged one that was completely dead and had some hydroids growing on it. But anyway, so Tina's not here today to help us identify the black corals, so she's doing her own travel. And we saw this earlier, Chris. You had stepped out, I think, to lunch. This looked to me like some kind of Farreid sponge.
Yeah, I think you nailed it again, Scott. I would agree. This is Farreid. You've got, this is in the family Farreidae. It is a glass sponge in the Hexactinellida group, as opposed to being a demisponge. And Farreids, first of all, they have very, very thin walls, so you can almost see right through them, or you can see right through them. And then the second thing is that they form these tubes-like structures, and you can see these round tube openings. These are osculum. And so this is also what we refer to as a hard sponge or a brittle sponge, because it glues the spicules or microscopic parts together to form a framework called a dictyonal framework. And so this is a beautiful example of a Farreid sponge, and I'd go probably as far as Farreia near or Reef Oka.
Thanks, Chris. I'm trying to figure out what that yellow thing is that's moving inside there. We saw that on the earlier sponges as well. At first I thought it was a worm, but then it started to move around. Suggested maybe it was some kind of crustacean, perhaps some kind of squat lobster. See another one of the Venus flytrap anemones there on the left. Lots of black coral, lots of the plexaurid fins, primnoid bushes, probably Narella. No shortage of coral in this particular area that we settled in. Really nice. That, yeah, that's a black coral that the Venus flytrap anemone is on right here. Earlier, the one we saw was on a Metallogorgia skeleton. So how did that rock collection go? You get something pretty good size?
We got some pretty good size. We'll see what it is when we get it up, but it was a good chance to get one. So it looks like we'll have some more opportunities here. There seems to be some loose stuff on the steeper part of the slope. So there was plenty of biology on it. So.
That 1,000 meter depth boundary is challenging for us today.
We've approached it a couple times. So.
Del, could you make a comment about what type of rock we're seeing here? Is this ancient reef or is it basalt? We had a question here from visitors.
Yeah, that's a good question. We're at fairly shallow depths here, at 1,000 meters now. Most of the larger seamounts that we see in this area that are above about 1,300 meters tend to have flat tops, which would tend to suggest that was all exposed or near the surface at one point. This is a little pointier on top. It's not really flat, this little nub that we're on. But I think what we're seeing here, first of all, the texture of the rock, the black color is all this ferromanganese crust that we've been seeing all over. That can actually make it a little hard to see what's here, but sort of the tabular look of some of this, yeah, it does look like it's old reef material to me. And one of the things we'll be able to check when we look at that rock later. But yeah, this looks more like reef material than lava. Of course, the whole structure underneath is built up on a seamount, so originally it was a volcanic structure.
I don't know if we're in exactly the same place as before, but we had a look at that Solandaria hydrozoan that was undergrowing a ledge. And there's a Metallogorgia colony again.
Yeah, we came down. It looked vaguely familiar. So
look at our track. Yeah, I don't know how far back it goes. That's very impressive. Very impressive navigation. Go ahead, Nat, watch lead. Copy that, Nav, thank you.
yeah, for folks on shore, we're just dealing with some operational issues with the tether again here. So please stand by, and we'll be underway again soon.
Nav, it's watch lead. Understood. Thank you for the update.
All right, so folks on shore, just stand by with us for 1 more minute. Video, this is survey. All right, folks, so you're going to be rebroadcast in just a second. If you want to talk through some wrap-up while I go confirm what's going on.
So hello again all. So we're getting our final views of the bottom here. We're still at the base of this cone, kind of tucked between two different cones on the surface of, I guess I would call this what, the lower slope. I don't know, Del, you were talking about this overall platform-like feature that Kingman is rising out of. So is this a plateau?
Yeah, we'd call this a carbonate platform, or a
carbonate
plateau.
platform.
Yeah.
Yeah, so we're at the base of one of these cones on this carbonate platform, and there's another one of these cones just to our east. And so it looks to me like currents are sort of sweeping through and being squeezed between these two cones, and it may explain why we have just incredible diversity and abundance of these corals fans at the bottom. And you can see their beautiful color, lots of different species, lots of different corals, some sponges. And we hadn't really even gotten onto the slope yet. And so I'm curious to know what this would have been like at the top, given what we saw here. But I can also say, we're talking about a little debrief here. I noted that there was a lot of similarities in terms of some of the fans compared to what we saw in Atlantic canyons. And I see Casey has come back. Oh, not talking to us. So does anyone on shore have any comments about what we saw?
I'd just like to say that this was a great dive for fish. We're going to be busy annotating all the fishes.
That's good. It was a great dive for corals and for fish. And we got a rock.
And we got a rock. Most important of all.
It's all your perspective.
Yeah, that rock had lots of biology, so Scott will be very busy this afternoon.
Wonderful. Wonderful. I love this small stuff. And the big stuff and the intermediate
It
stuff.
was interesting to note the abundance of fish here, even with the short dive, compared to yesterday when we were in such a low oxygen area. Oxygen wasn't high here, but we certainly saw many more fish per square meter, well, not square meter, but per area, and greater diversity of fish on this dive. And there goes another one through the D2 view, probably a midwater species. But so it was interesting. It is just disappointing that we couldn't get to the top, but that's the nature of exploration. You know, you never know how it's going to go.
Thank you, Bruce. And Chris, I'm not sure if you're still on the line. I see you're not in the chat room anymore.
I'm still here.
All right.
I liked what we saw at the first. It would have been nice to get to the summit, but I guess that's where I'm at. But what we did manage to image was really cool. We're seeing a really nice mix of corals and different fish. So that's really good, and look forward to the dive tomorrow.
Thank you, Chris.
All right, guys, this is Casey. I'm back. So we're going to do the dive planning call at 2 p.m. SST, which means that is 3 p.m. Hawaii, 6 p.m. on Pacific, and 9 p.m. on the East Coast. So we will send out an email to update you guys to that. But something that I think may be worth considering and thinking about over the next hour or two is we have one more dive in this area tomorrow. Do you want to try and come back to this site and try and get up to the surface? Or do you want to potentially try for another cone as we have planned? So something to think about.
By surface, you mean the top of the cone?
Yeah, the top of the cone. I'm sorry.
Sure.
Yeah,
I'm glad you raised that possibility, because I do think that's something that everyone should consider. And this obviously was a great start to it, and maybe that's more intriguing than going down to 2,000 meters. So.
Right, and we could potentially pick the other side and come up and go to the summit. So
sure.
There's we have the flexibility to adapt based on the conditions. So if you guys think about that over the next couple, like hour and a half or so, and we will chat again at 2 p.m. here on the ship, 3 p.m. Hawaii, 6 p.m. Pacific, 9 p.m. Eastern.
We're all over the world, folks. Thanks for joining and helping us out, folks.
Before we sign off, does anyone have any questions? All right, thank you, everyone. Talk to you soon.